Students, in biotechnology and breeding and biotechnology lesson, we are going to see about genetic engineering. What is genetic engineering? Genetic engineering is the manipulation and transfer of gene from one organism to another organism to create a new DNA called as the recombinant DNA or rDNA. So basically, genetic engineering, the term is the recombination. That is the combination of two different DNAs from two different sources are joined together. So only it is called as genetic engineering or recombinant DNA technology. So in this technology, they are using the important is the discoveries that leads to the stepping stone of RNA or DNA technology. That is the presence of plasmid in bacteria is replicated indefinitely with the chromosomal DNA. The help of a restriction DNA cuts or the breaking of a specific site has been made. So DNA ligase are the enzymes which helps in joining or ligating the broken fragments or the DNA fragments. So DNA ligase is an enzyme. With the help of this, two different DNAs are joined together and they are creating a new DNA, that is the rDNA or recombinant DNA. Now let's see what is gene cloning. So human gene cloning or commonly called as a gene cloning. So here the same individual copy of a DNA is again synthesized, or the carbon copy of an individual is often called as clones, or the technique involved in this is called as the cloning, gene cloning. So here in humans, what they used to do in gene cloning, the genes or a piece of the DNA fragments is inserted into a bacteria where the DNA will be multiplied. Okay, so if you are need of a particular DNA sequence, that sequence is collected and they are inserted into a bacteria. So it is having a special structure called as the plasmid, and there it makes many copy of it. Okay, the same picture of the same copy which we have inserted is taken more and more, then they are transferred into the particular genes. Okay, so the first step in gene cloning, or in gene cloning, many brief technique or outline of this basic involvement in gene cloning is isolation of the desirable fragments using restriction enzyme. So restriction enzymes are used to isolate the DNA fragment. Then isolated fragments are inserted of the DNA fragment into the suitable vector. After isolating from the human, they are inserted into the plasmid or the vector, that is the bacteria. Bacteria acts as a vector here. So we are getting a DNA fragment from a human being and we are inserting it into a bacterial DNA where they are joining. So transfer of rDNA into the bacterial host is called as the transformation. So after this, selection and multiplication. So we have selected, now we have recombinant, that is we have jointed the both the DNAs, that is a bacterial DNA and a human DNA. So that on the whole, the joining chromosome is, or the DNA is called as a recombinant DNA. Now this recombinant DNA is inserted into the bacteria where selection and multiplication is taking place inside the bacteria. So inside the bacteria it goes on division. So the number is increasing. Then finally expression of the cloned DNA in the host. And finally the whole DNA will be seen. So after cutting, so all restriction and isolation has been done outside in the vitro condition, and they are inserted into the bacteria. After inserting it, the bacteria will take many clones of it, and finally it expresses the clone gene into the host cell. Now this can be used for the various uses. Using these strategies, several enzyme, hormones and vaccines has been produced, okay, with the help of genetic engineering. So genetic engineering, only isolation is the main technique. So we are in need of a particular DNA, means that particular part is alone removed. Okay, with the help of restriction enzymes, we are cutting. So restriction enzymes helps in cutting or breaking down of the DNA strands. So we know DNA is having a double helical strands. So this strands has been cut with the help of the restriction enzyme, or it is also called as the molecular scissors. So restriction enzymes is otherwise this is one of the important one word, okay, which is termed as molecular scissors. That is restriction enzymes are termed as the molecular scissors, which is cutting the DNA sequence. So that's all about gene cloning and genetic engineering in human being. So we shall see biotechnology in the field of medicine in the next class. Thank you.